Hello, my name is Gonzali Onder, and I spent the summer working in Professor Eniola's Targeted Drug Delivery Lab at the University of Michigan. The goal of our lab is to improve and create new drug delivery methods. For a drug to be most effective, it needs to have three things: a carrier, a way to target a specific site or population of disease cells, and a way to be absorbed. For example, let's consider a chemotherapy agent administered to a cancer patient. In this case, the carrier of the drug is the solution that the drug is in. The drug gets to the cancerous area by being distributed in the bloodstream to all the cells of the body, and is absorbed non-specifically by all the cells that it touches. Many cancer drugs target and minimize cell division, which minimizes tumor growth. However, since all cells contain DNA, these drugs are just as poisonous to the rest of the body as the cancer cells. This limits the amount of the drug that is able to be administered without harming the patient. If only there was a way to target the cells that needed the drug to simultaneously maximize efficiency while minimizing side effects. Our lab aims to solve this problem by utilizing nano and micro-sized particles as drug delivery carriers. The goal of these particles is to deliver the drugs to only where they're needed and release the drug there, rather than non-specifically to the entire body. Our lab's goal is to target heart disease and the inflammation caused by arterial plaque. However, we cannot just inject these particles into humans to test them. Therefore, after fabricating these carriers, our lab had to create a model to test the ability of the particles to adhere to the location that they are needed at and release their drug. It is well known that heart disease and plaque buildup affects the cells in the innermost layer of blood vessels, also known as endothelial cells, and causes inflammation. This inflammation then causes an increase in the expression of the protein E-selectin, a protein found in the extracellular matrix, or ECM, of the cell. For this reason, in our model we used human umbilical vein endothelial cells that overexpress E-selectin. We isolate cells from donors and then culture them in plastic dishes and glass cover slips. Using a protein called IL-1 beta, we can then cause the cells to overexpress E-selectin, and after placing the particles into various solutions and perfusing them over a monolayer of activated cells, test the adhesion efficiency. Our aim by culturing these cells and running solution over them is to create the most physiologically relevant model. However, many different factors can affect E-selectin expression. For example, a paper from 2012 titled "Shear Stress Modulation of IL-1 Beta-Induced E-selectin Expression of Human Endothelial Cells" shows that E-selectin expression is dependent on time of exposure to IL-1 beta and the shear rate at which it is run over the cells. For example, when IL-1 beta is perfused at 5 dynes per centimeter squared, there are twice as many E-selectin sites per micron squared as when it is perfused at 1 dyne per centimeter squared. Another factor that has recently been brought to light is the effect of material modulus. Research has been done that suggests that cells express proteins differently depending on the stiffness or modulus of the material on which they are cultured. Endothelial cells in the body grow on smooth muscle cells, which have an elastic modulus on the scale of 10 to the 6 pascals, whereas glass, the material which we currently grow our endothelial cells on, has an elastic modulus on the scale of 10 to the 10th pascals. This implies that the cells in the glass, when exposed to IL-1 beta, may not be expressing E-selectin at the same levels as in the body. My project for the summer was to begin developing a new surface for endothelial cells to grow on, one that had a modulus more representative of the 10 to the 6 pascal modulus of smooth muscle cells. To create this more deformable surface, we began exploring the option of hydrogels. Hydrogels are gels composed of a usually polymeric backbone that contain a large percentage of water. They have been used for many different studies on the effects of modulus on protein expression, as their modulus can be modified by changing the concentration of water or precursor, or the molecular weight. We specifically decided to use polyethylene glycol, or PEG, as our backbone polymer, as PEG is very hydrophilic due to its structure. Using these two modified PEG polymers, we were able to design a photo-initiated and radical-mediated polymerization reaction to allow the PEG to self-assemble into the backbone structure of the hydrogel. We then tested the variation of modulus we could achieve with the PEG hydrogels using an ARG2 rheometer. This specific rheometer measures shear modulus by measuring the stress and strain on the hydrogel. After the precursor material is injected into the machine in between two plates, a UV light is shined on the precursor, activating the reaction, while slightly rotating one of the plates. Stress and strain measurements are then taken on the material, which gives modulus. However, before the protein levels of cells growing on these gels can be tested, the cells must be given a way to attach. Cells adhere to surfaces through the interaction of their ECM proteins with proteins found on the surface. However, PEG repels proteins and is routinely used to protect polymer particles from protein opsonization. Since the cells would have difficulty growing on PEG alone, the next step for the project is to functionalize and incorporate the necessary proteins into the radical-mediated network. These proteins will then allow endothelial cells to grow into the hydrogel so that the expression levels of the proteins we target can be reassessed under flow conditions, further improving the lab's blood vessel and heart disease model and allowing us to more realistically test the functionality of targeted carriers. Thank you for listening.